Good day wherever you are. Welcome to today's lesson, and today we are dealing with Kcse 2014 practical biology, and let's see what the students were asked. In question 1, you are provided with solutions labeled Q and R, a substance labeled D, and a delivery tube fitted with a rubber band. Then you are told, label solution Q as the lime water. You know lime water is calcium hydroxide. Then label solution R as 10% sugar or sugar solution. Add substance D to the 10%. Now the substance that you label D to 10% of sugar solution. Tightly close or plug the boiling tube with the rubber band fitted with the delivery tube. And then dip the other end of the delivery tube, the test tube containing lime water. Put the boiling tube in the warm water bath at 40 degrees Celsius and allow the setup to stand as shown in the diagram below. Then observe the setup for about 15 minutes. Now the setup was looking like this. So you can see this is the lime water, the test tube, delivery tube, cork, substance D, and the sugar solution mixture under the water bath of 40 degrees Celsius. Then you are told in question 1, state the observation made in the lime water. Now the observation was a white precipitate. White precipitate was observed, or effervescence. Effervescence occurred. Yes, it was the observation. Now explain the observation made in lime water. The observation was, now here in this reaction there was a gas that was produced, and you know that is carbon 4. Now the carbon 4 reacted with the lime water to form the white precipitate. Now we will say the gas produced is carbon 4 oxide, which reacted with the lime water, to form the white precipitate. Name the physiological process that was being investigated. Now this process was respiration. Write a word equation for the physiological process investigated. Now the word equation will be glucose, or the sugar that we have, the 10% sugar, glucose plus oxygen, and in the presence of yeast or the enzymes, to give you carbon 4 plus water as energy. Yeah, that was the word equation. This is carbon 4 oxide. Carbon 4 oxide should be able to differentiate between a chemical equation and a word equation. Roman 5, it says, why was the warm water bath used in this experiment? Whenever you see something is being warmed and then there is presence of enzyme, automatically you should know that it was to provide optimum temperature for the action of the enzyme. Yes, and then in B it says put a drop of the content in the boiling tube on the microscope slide. Now here they are provided with microscope, and then stain with a drop of methylene blue and cover with a cover slip. Observe it under the light microscope using the low, medium and the high power objective lenses. Draw and label one of the structure observed under the high power of the objective lens. Now, for us, we don't have the microscope, but I will do for you the expected observation. You would see a structure looking like this. Structure of the plant cell. Then there is a nucleus. This is the nucleus. The outer layer is the cell wall. Then the inner is the cell membrane. And the aim of the fluid like this is the cytoplasm. Now state the magnification of your drawing. The magnification, you will multiply the eyepiece lens, you multiply by the objective lens in order to get the magnification, which is times 400. The answer should have a times sign, a multiple sign. Identify substance D. State and identify the substance D. Now here, substance D was the yeast. Question 2. You are provided with the specimen labeled E and F. E and F, these had the network veins and had the broad leaves like a bean, and this one had parallel veins like for maize. Now this from class Dicotyledonine, this from class Monocotyledonine. That was, I observed that. Then name the subdivision to which the specimen belong. After you have known these classes, you should know the subdivision. Now the subdivision was the angiospermatophyte or angiospermaphyte. Angiospermatophyte. Angiospermatophyte or angiospermaphyte. Now use observable features of the specimen. Give 2 reasons for your answer. You'd say they has parallel veined leaves, then have flowers. These are the features you should observe. State the difference between the leaf of specimen E and F. Now the differences were E had opposite, their leaves were opposite, while in F their leaves was alternate. In E there were network veins, and they were net veined, and these were parallel veined. And here had serrated margin, while F had the smooth margin. Here this was green, here this was the purple. Has this head petiole, has petiole, this one had the sheath, was modified into the sheath. But now the difference between specimen E and F, the stems of the specimen E and F. The specimen of E was green, while for F was purple. While the other thing, this was thorny and this was smooth. Then C, using observable features on the specimen, state the adaptation of the stem of specimen E to its habitat. Now you have seen E had thorns. Now from there we will say E had thorns, had thorny stem for protection against herbivores, so as to survive herbivores. That's one. The other one, its stem had a hard stem for mechanical support, to support it in order to survive in its habitat, for mechanical support. In question 3, it says the photographs below shows two skeletal ribs of a mammal. Is A and B. This is the A, this is B. Now before answering question, let's look the structure as well. As you can see, this is the scapula. This is the humerus. So therefore, this whole skeleton, this is the forelimb. This is from in the arm, it is known as the forelimb, and in the leg, this is the hind limb, because this is the femur. We have tibia and fibula. Yeah, here we have ulna and radius. I hope it is visible, you are able to see. Now let's answer our question after we have identified the skeletons. Which of the two skeletons represent a forelimb? As you have seen, A, this is A, this is B. A, this is the forelimb, A. Give reasons. Roman, to state the features observable on the skeleton to confirm your answer. We have seen it has the scapula. There is presence of scapula. Also, there is presence of humerus, presence of ulna and radius. You can write those ones. Presence of humerus, presence of ulna and radius, ulna and radius, and etc. Now let's go to B. Name the bones labeled J, K, and M. Now J is the lower, K is here, K, this is J and K. Then M are here, M are the hind limb. Now J J, as you see, the first one is the ulna, the second is the radius. Therefore, J is the radius. K, then K is here. K, this is the femur. K is the femur. And here we see this is the tarsals, metatarsals, this is the phalanges. M is the phalanges, this one. Here for M is the phalanges. Yes. Which bone forms the second joint with the bone labeled K? Now K is the femur. Which bone forms the second joint? Now we are asked about the bone above K, above the femur. You know that is the pelvic girdle. And then D, you are asked, name the type of joint formed at the part labeled H and L. Part labeled H. Now H, as you can see, is here. Is the H, the wrist. Here is the wrist, and here between femur and the tibia and fibula. Now you are asked to name the joint. Here in H, in the wrist, this is known as the gliding joint, while in L, this is the hinge joint, because it can only move in only one plane, cannot move at 360 like the ball and socket joint, which is found there above the femur. Therefore, H is the gliding joint, while L is the hinge joint. Then lastly, it says, apart from the bones, state the function of any two other components of a joint. Now you should be able to know the components of a joint. There are many, but let's give like 3 or 4. Let's give 4. First component is the ligament. Ligament, its function is to attach a bone and another bone, a bone to another bone to another. While another one is the tendon. You should be able to know difference between this one, ligament, and tendon. Tendon attaches a bone and a muscle, while this one attaches a bone and a bone. The second one is the cartilage. Cartilage is found in the vertebral column. The cartilage acts as a cushion to reduce friction. Here, function is to reduce friction. Another component of joint is this synovial fluid. That is a fluid found in the joint. This one lubricates also by reducing friction. Lubricates by reducing friction. Another one is the synovial membrane. Synovial membrane, this one secretes the synovial fluid, secretes the synovial fluid. That's all. Thank you. Now this is Kcse Biology Practical 2015. Let us enter and see what it contains. The first question, a student is required to spend 15 minutes in reading the instruction First question, as you see, is from dichotomous key. You are required to fill the blank space you are provided. The second question is from bounds. You are required to study them and see the question you are given. Identify the question carefully, and lastly, question 3. Question 3 is for food test. As you see, you are provided with solution D, 4 test tubes, iodine, and Benedict's solution. Now let's start with question 1. In question 1, as you have seen, it says, using the pictures of animals provided below, complete the construction of the dichotomous key by filling the blank spaces. This dichotomous key is from topic in classification. Students should be able to identify the distinguishing characteristics for each organisms. Identify class, the kingdom, and the characteristics and the phylum. Let's start with our question. In A, we say animals with backbone and animal without backbone. Come back to the pictures. Animals with backbone are those from phylum Chordata, because they have a unique characteristic that says they possesses a notochord, which is the backbone. You should be able to identify them. They are the eagle from class Aves, fish from class Fishes, tortoise and frog, and the not the tortoise, is the frog. These 3, class Aves, class Fishes, and class Reptilia, they have a notochord. Now you will say the 3 have a backbone. Now we are told to go to 2. Those without backbone, you leave that like that. And then you have seen, we have identified the 3. Now you look at distinguishing characteristics of the 3. Like now we will say animals with wings, animals without wings. Now animals with wings is eagle. You write eagle. Animals without wings, you leave like that. First go animals without wings, you say go to 3. When you go to 3, you distinguish these 2, the class Fishes and class Reptilia. You say animals that live in water for long or for all time, that live in water for all time, animals which live in water for some time. That the animal live in water for long is fish. But here it's not only the tortoise live in water, also the starfish and the octopus. They live in water but not for so long. Now you write go to 4, and then you say even this live in water, but now you distinguish this one and this one because they live in water for some time. I will say animals with scales, animals without scales. Animals with scales is tortoise, animals without scale is frog. Now you have identified this one, you have classified it, you have classified this one, and this one. Now we are remaining with earthworm, starfish, spider, and octopus. You come to 1A, you didn't fill the space for B. Now because we have reached 4 and we are supposed to go 5, you say here go to 5. When we go to 5, animals with legs, animals without legs. Here for animals with legs, animals without legs. Here, because there are 2 with legs and 2 without, you write go to 6, because it's only one characteristics that can only satisfy an organism, not 2. Now because you have gone to 8, you say now for 8 go to 6. Say animals with 8 legs, animals with a shell. Now this is very easy because animals without legs we have been told to go to 7, but here with legs we have been told to go to 6. Now when we are at 6, animals with 6 legs have been given butterfly. Animals with 8 legs, that automatically you should know that the organisms of class Arachnida have 4 pairs of legs. So you should know therefore that is spider. And now we have finished for go to 6. Now animals without legs go to 7. Animals with shell we have been given is snail. Animals without shell, we see snail have, but we have classified this one. Now we are remaining with starfish, octopus, and earthworm. Animals with shell we have been given snail. Without shell they are 3. Now you say go to 8. Go to 8. In 8 we have been told animals with jelly-like body and animals without jelly-like body. Now animals with jelly-like body is there. Animals without jelly-like body you can see is the starfish. Don't have this, but this one to have jelly-like body. Now you say here with jelly-like body, the 2 have jelly-like body, there's earthworm and the octopus. We say go to 9. And animals without jelly-like body we say starfish. Because we are remaining with the 2, this will be easy because octopus and earthworm, you say animals with segmented body, animals without segmented body. Automatically you see this are the earthworm is segmented into spaces, small spaces. Now you write automatically is earthworm. That's how to answer a dichotomous key. And in other question we look at, a student is given to construct all the dichotomous key. You should be able to know how to construct your own dichotomous key without being provided with the steps to follow. Now we go to question 2. Now in question 2 it reads, below the pictures of 3 mammalian vertebrae. Now here a student should be able to know how many vertebrae human beings have. You know we have cervical vertebrae, lumbar vertebrae, thoracic vertebrae, caudal vertebrae, and sacral vertebrae. Those are 5. Now, for those 5, you should have the unique characteristics that you should be able to identify. Now, let's start identifying ours. The first F is cervical vertebrae, because it has a wide neural canal. It's wide, very wide, as you see, and also it has vertebral canal. Now that is F. You have identified it is cervical vertebrae. G, this is thoracic vertebrae, because this has many extended projection. That's why you say this thoracic vertebrae. This cervical is found in neck region, and this one is found in thoracic region. Now we go for H. This is lumbar vertebrae, because first of all it has long neural spine, and also has a wide and large centrum. Now let's go to the question. After you have identified the bones, now you go to the question. Identify the type of vertebrae labeled F, G, and H. As now your work will be easy because you have identified. Now F is cervical vertebrae. F is cervical vertebrae or vertebra, and G is thoracic vertebra, and H is lumbar vertebra. Now you are told now in B, label 5 parts of the vertebra labeled H. H, you know now is lumbar vertebra. Now you should be able to label. Now we start. The first one, this is neural canal. Here, this is metamorphosis. This is transverse process that we have been told to label 5. This is first, second, third. Now we have labeled the fourth, which is the centrum. Now the fifth, neural canal. Now you get your 5 marks. Now after you have labeled B, now you go to C. Name the articular facets labeled K and L. K and L are found in thoracic vertebrae. Now you should be able to know that K is tubercular facet, and L is capitular facet. S is tubercular facet, and L is capitular facet. After you have identified, now you name. name in your question. This is tubercular facet, and this is capitular facet. Now we proceed. How does each of the parts of the vertebra enable a mammalian skeleton to carry out its function? Now this is where you should come and identify the parts and the function of those projection and this centrum. Those are things that you are being asked. Now for the first one, you can say it possesses a large or broad centrum to support the body weight of the vertebrae. Now you see we have large or broad centrum to support the body vertebrae. That's the function of the centrum, is offers support to the body of the vertebrae. Now the second one, as we see, they have the second one, as you see, all of them have the neural canal. Now you say have neural canal to allow for the passage of the spinal cord. Finish. Next we have the possesses, as you see, all of them have extended projection. I will say they have this extended projection and the transverse process. We will say they have transverse process which helps in attachment of the abdominal muscles. A learner should notice that and know that all projection in any body, any projection offers a surface for muscle attachment. Now you go and see that they have facets. You see, have facets for articulation with other vertebrae. Now we have answered our 4 marks. The first, we have centrum, neural canal, transverse process, and facets. Now number 3, our last question is on food test. You are provided with a 250 beaker, 4 test tubes, solutions labeled D and E, iodine and Benedict solution. First of all, you should be able to know that the reagents you are provided are used to test for what? Iodine is used to test for starch, Benedict solution reducing sugars. Now we read the procedure. Half fill the beaker with the hot water provided to create hot water and water bath. Now 1. Label the 4 test tubes as follows. Test tube 1, D plus iodine solution, that is solution D and iodine solution. Roman 2, you test in test tube 2, D plus solution D plus solution E plus iodine solution. And Roman 3, in test tube 3, solution D plus Benedict solution. And Roman 4, test tube 4, solution D plus solution E plus Benedict solution. In 2, they are saying put 1 centimeters cubed. Put 1 centimeters cubed of solution D in each of the 4 test tubes. This also means 1 milliliter. Put 1 milliliter of solution D in each of the 4 test tubes. In Roman 3, to the solution D and iodine in test tube, add 1 drop of iodine solution. This means now that you are in test tube 1, you labeled D plus iodine. Test tube that, add 1 drop of iodine solution and shake to mix. Therefore you should observe what goes on. In Roman 3, to the test tube that you labeled D plus E plus iodine, that is D plus E plus iodine, test tube 2 is test tube 1, test tube 2.
Don't forget to subscribe. Just a button, press the button, and that's all I need.
That you labeled test. This one is test tube 2. Add 1 centimeter of solution E that you provided and 2 drops of iodine and shake. To test tube that you labeled D plus iodine, that is test tube 3. Add 1 centimeter of Benedict solution, shake and mix. Shake to mix. Now in 4, to the D plus E plus solution test tube, that is test tube 4. Add 1 centimeter of solution E and 1 centimeter cubed of Benedict solution, shake to mix. Now after you have done that all, observe the changes in each of the 4 test tubes. After you have observed in each, put all the 4 test tubes in a hot water bath and observe carefully for about 5 minutes. In practical, even in chemistry, is only following the procedure and you will understand. Let's go to the questions. Now here we are lucky because we are not told to write the procedure. We are only told to write the observation and conclusion. Now a student is able to know what is testing because you have been given starch, iodine, and reducing sugar and Benedict. Therefore, students should be able to understand this testing for reducing sugar and starch. After in test tube 1, after you have done as he was instructed, the observation should be a black solution is observed. Now conclusion automatically is starch present. Tube 2, as we are told, you put solution D and E and iodine solution. Now your observation should be a colorless solution is observed. This simply means that the starch was absent. Now in 3, the solution D plus Benedict's solution. This means that your observation will be the blue color of Benedict's solution remained blue. This shows that reducing sugar was absent. That is the conclusion. In test tube 4, you have put solution D and E plus the Benedict solution, and you have observed that an orange color is observed. That is the observation observed. Now automatically reducing sugar present. In B, you are told what is the role of each of the following in the experiment. The role of solution E was to hydrolyze starch, and the role of hot water bath was to activate the enzymes. So now you need to have knowledge in nutrition and in animals. Give the identity of E in human beings. Now you should be able to know that the enzyme that breaks starch, that is the salivary amylase. Or ptyalin. Now D. Explain the observation on the reagent tested with Benedict solution. Now that is your own observation. You should record your own observation. The paper is over. That was what was needed by the student in 2015 to get all the marks. Thank you.